Good day, this is Professor Will, CBMG PhD. And today's Wednesday, January 12, 2022. It is 1:49 and 25 seconds, p.m. Eastern Standard Time. And today's research, electric fields. So let's hop to it here. Using only 100 atoms, electric fields can be detected and changed. Date January 11, 2022. Source, University of Southern California. The body is full of electrical signals. Researchers have now created a nanomaterial that is capable of both detecting and modulating the electric field. And this new material can be used in vitro studies for reading and writing the electric field without damaging nearby cells and tissue. In addition, researchers can use this material to conduct in vitro studies to understand how neurons transmit signals, but also to understand how to potentially shut off errant neurons. This may provide critical insights on neurodegeneration. In addition, there is a growing body of evidence that recording and altering the bioelectric fields of cells and tissue play a vital role in wound healing, and even potentially fighting diseases like cancer and heart disease. Now, for the first time, researchers have created a molecular device that can do both record and manipulate its surrounding bioelectric field. The triangle-shaped device is made of two small, connected molecules, much smaller than a virus, and similar to the diameter of a DNA strand. It is a completely new material for reading and writing the electric field without damaging nearby cells and tissue. And each of the two molecules, linked by a short chain of carbon atoms, has its own separate function. One molecule acts as a sensor or a detector that measures a local electric field when triggered by a red light. A second molecule, the modifier, generates additional electrons when exposed to blue light. Notably, each function is independent, controlled by different wavelengths of light. Though not intended for use in humans, the organic device would sit partially, inside and outside, the cell's membrane for in vitro experiments. The research laboratory was responsible for creating the new organic molecule, while the team played a key role in testing how efficiently the modifier was generating electricity when activated by light. Because the reporter molecule can insert into tissue, it has the possibility to measure electric fields non-invasively, provide ultra-fast 3D high-resolution imaging of neural networks, and this can play a crucial role for other researchers testing the effects of new drugs or changes in conditions like pressure and oxygen. Unlike many other previous tools, it will do so without damaging healthy cells or tissue or requiring genetic manipulation of the system. And this multifunctional imaging agent is already capable with existing microscopes, so it will enable a wide range of researchers from biology to neuroscience to physiology, and to ask new types of questions about biological systems and their response to different stimuli, drugs, and environmental factors. The new frontiers are endless. In addition, the modifier molecule, by altering the nearby electric field of cells, can precisely damage a single point, allowing future researchers to determine the cascading effects throughout, say, an entire network of brain cells or heart cells. And if you have a wireless network in your home, what happens if one of those nodes becomes unstable? How does that affect all the other nodes in your house? Do they still work? Once we understand a biological system, like the human body, we can better predict its response, or alter its response, such as making better drugs to prevent undesirable behaviors. Researchers say that they can use this to both interrogate as well as manipulate, and they can do both things at very high resolutions, both spatially and temporary. The key to the new organic device was the ability to eliminate crosstalk, how to get these two very different molecules to join together and not interfere with each other in the manner of two scrambled radio signals. In the beginning, it wasn't entirely obvious that it was even going to be possible. The solution: separate both by a long alkyl chain, which does not affect the photophysical abilities of each. The next steps for this multifunctional new molecule, including testing on neurons and even bacteria. Researchers previously demonstrated the ability of microbial communities to transfer electrons between cells and across relatively long distances, with huge implications for harvesting biofuels. Good day, this is Professor Will, CBMD PhD, on microbiology, bioinformatics, and pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.